Hello, my dear students. Welcome to a new episode of biology. Today, we will study a new part of science, which is the cell cycle. The cell cycle is the life cycle of the cell because the cell is like any living thing that has its own life cycle. So, let's start by the key concept, which is cells have distinct phases of growth, reproduction, and normal functions. The main ideas number one, the cell cycle has four main stages, and we will study them all. Number two, cells divide at different rates. Number three, cell size is limited. So let's start. The cell cycle has four main stages. Just as all species have life cycles, cells also have their own life cycle. The cell cycle is the regular pattern of growth, DNA duplication or the copying of the DNA, and the cell division that occurs in eukaryotic cells. Recall that eukaryotic cells are the cells that have a nucleus and a membrane-bound organelles. The four main stages of the cell cycle are gap 1 or G1, then synthesis or S phase, then we have gap 2, which is G2, and mitosis. Gap 1, synthesis, and the gap 2 make up the interphase. As you can see, this is gap 1, synthesis, and the gap 2. The three of them make up an interphase. Then we have mitosis, followed by cytokinesis. Okay, the stages of the cell cycle get their names from early studies of cell division. The scientists' observations were limited before by the microscopes of the time. When a cell was not actively dividing, they couldn't see activity in it. Thus, they originally divided the cell cycle into only two parts: the interphase, when the cell appeared to be at rest, and the mitosis, when the cells started to divide. Then improved techniques and tools later allowed scientists to detect the copying of DNA. This is the S stage DNA synthesis, and they changed their description of the cell cycle to include the synthesis stage. Then scientists named the periods between mitosis and the synthesis gap 1 and gap 2 because they still couldn't see anything happening during the other parts of the interphase. Then eventually the scientists learned that during interphase, cells carry out the normal functions and undergo critical growth and preparation for cell division. Okay, so let's talk about the phases of the cell cycle each. Gap 1 or G1, this is the first stage of the cell cycle. This is gap 1. During gap 1, a cell carries out its normal functions. If it is a skeletal muscle cell, it would do its normal job, which is contracting to move joints. And if it's an adrenal cell, it secretes hormones such as adrenaline. If it is an intestinal cell, it will absorb nutrients. During G1 or gap 1, cells also increase in size and the organelles increase in number. This is so important. A cell spends most of its time in G1 stage, although the length of this stage varies by cell type. During G1, the cell must pass a critical checkpoint before it can proceed to the synthesis stage. So there are some checkpoints that must be passed before the cell moves from G1 to S stage. Just as it would be dangerous for you to run a marathon if you had not slept or eaten for several days, it would also be dangerous for your cells to continue dividing if certain conditions were not met. For instance, most animal cells need enough nutrition, adequate size, and a relatively undamaged DNA to divide successfully. They also need specific signals from the other cells, telling them whether more cell division needed or not. So, in case of not having undamaged DNA or there is not enough nutrition or the cell have reached the adequate size, so the cells would not divide successfully. S stage or synthesis. The second stage of the cell cycle is the S or synthesis. Synthesis means the combining of parts to make a whole. During the S stage, the cells make a copy of its nuclear DNA. In this case, in eukaryotes, the DNA is located in the nucleus. During interphase, the DNA is loosely organized and appears grainy in photographs. But by the end of the S stage, The cell nucleus contains two complete sets of DNA, so the copying is done. Then the third stage, which is gap 2 or G2. During G2, cells continue to carry out their normal functions and additional growth occurs. So the cells grow larger in size. Like G1 stage, it includes a critical checkpoint. Everything must be in order, adequate cell size, undamaged DNA before the cells goes through mitosis and division. Then we reach mitosis. Mitosis or M stage is the fourth stage of the cell cycle and includes two processes, mitosis and cytokinesis. Mitosis is the division of the cell nucleus and its contents. During mitosis, the nuclear membrane dissolves, then the duplicated DNA condenses around the proteins, then separates, and the two new nuclei form. Each one of them have a complete set of the DNA. Then lastly, cytokinesis is the process that divides the cell's cytoplasm. This is so important. The result is two daughter cells that are genetically identical to the original one. So mitosis divides the duplicated DNA, it divides the nucleus, and cytokinesis divides the cytoplasm. Okay, the stages of the cell cycle and the proteins that control it are similar in all eukaryotes. You might be surprised to know that scientists have demonstrated that some of the molecules that regulate checkpoints in the yeast cell cycle can also work in human cell cycles. Such similarities suggest that all the eukaryotes share a common ancestry. Then the cells divide at different rates. As you can see from this table, we have the skin cell. This is the table to tell the cell type and the approximate lifespan. Skin cells, it divide or its lifespan is 2 weeks. Red blood cells, 4 months. Liver cells from 300 to 500 days. The internal lining of your intestine will be from 4 to 5 days, but the muscles and other tissues in your intestine will take 16 years. So the rates of cell division vary widely. In the prokaryotic cell cycle is similar but not identical to the cell cycle of eukaryotic cells because it is much simpler. Recall that prokaryotes do not have a membrane-bound organelles or cytoskeleton which is found in eukaryotes. So prokaryotic cells typically divide much faster than do eukaryotic cells. Yes, for sure. The rate at which your cells divide is linked to your body's need for those cells. So the rate or the speed of cell division inside your body depends on whether your body needs these cells right now or not. In human cells, the SG2 and the mitosis stages together usually take about 12 hours, but the length of G1 stage is the one that differs most from cell type to cell type. The rate of the cell division is greater in embryos and the children than in adults. Yes, because they are still building their bodies and their tissues and organs. Children have a shorter cell cycle and many of their organs are still developing. This is why the division rate is higher in children. But the rate of cell division also varies within different tissues of the adult body. How? The internal lining of your digestive tract receives a lot of wear and tear. It is unusual facing the hydrochloric acid and all the food that you eat, so it do a lot of effort. It receives a lot of wear and tear. As a result, cells that line your stomach and intestine are replaced every few days. In contrast, cells that make up the rest of your intestine, mainly smooth muscles, and many of your internal organs, such as lungs, kidneys, and liver, divide only occasionally in response to injury or death. So the smooth muscles that make up the rest of your intestine or other organs, such as lungs, liver, and kidney, they divide only in case of injury or death to repair the lost part of cells. Cells that divide only rarely are thought to enter a stage that some scientists call gap zero or G0. So what happens during gap zero? Cells are unlikely to divide, although they continue to carry out their normal functions. Some cells, such as neurons, appear to stay permanently in G1 and G0 stage. However, some data suggested that neurons actually can divide. They can divide, but they do not. This question continues to be actively researched. Other cells, such as lymphocytes, this is a type of your white blood cells, may remain in G0 for years until they recognize an invader, a pathogen, or a foreign particle inside the body, like bacteria, and so on. In this case, once the invader binds to a lymphocyte receptor, the lymphocyte goes through rapid cell divisions to help fight the infection. Then let's talk about another important part, which is the cell size is limited. Yes, the cells have an upper and lower cell size limits. If the cells were too small, can you imagine a cell too small? They couldn't contain all of the necessary organelles and molecules. For instance, a cell with too few mitochondria would not have enough energy to live. However, the cells cannot grow beyond a certain size, even if they are surrounded by plenty of nutrients. Why? The upper limit on cell size is due to the ratio of cell surface area to volume. This is so important. Recall that oxygen, nutrients, and waste more move across the cell membrane or the surface of the cell. Yes, these materials must be transported at adequate amounts and with adequate speed to keep the inside of the cell functioning, to keep the cell alive. So, but as the cell increases in size, its volume increases faster than the surface area, as shown in the next figure. I'll show you how. Therefore, further increase in size could result in a surface area too small for the adequate exchange of materials. In this case, as the cell grows, its volume increases more rapidly than its surface area. So, when the surface area to volume ratio is too small, the cell cannot move materials into and out of the cell at the sufficient rate or in sufficient quantities. Look at this table. It's okay, don't be afraid. So this table tells us, now suppose that we have three cells. The first one have a size of 1, and the second one 2, the size of one side, and the third one a size of 3. So these are three different cubes as three different cells. Now let's calculate the surface area and the volume for each one of them. Now, if the surface area for one side is 1 times 1, and we have 6 sides, so this will be 1 times 1 times 6 equals 6. For the second one, 2 times 2 equal 4, times 6 equal 24, 3 times 3 equal 9, 9 times 6 equal 54. Okay, this is for the surface area. So let's calculate the volume, calculate the space inside each cube of these. So this is the length times width times height. 1 times 1 times 1 equal 1. 2 times 2 times 2 equal 8. 3 times 3 times 3 equal 27. So as you can see, as the cell's volume increases, its surface area increases much faster. So let's calculate the surface area to volume ratio. 6 by 1, or this is equals 6 to 1. For the second cube, it equals 3 to 1. For the third cube, equals 2 to 1. As you can see, 6 to 1, this is the larger surface area to volume ratio. So this will be the best one. So the surface area is greater than the volume of the cell, so it will continue to take in the nutrients and to get rid of the wastes in adequate rate at adequate amounts for the cell to keep it functional or to keep it safe. Okay, then let's move on. Some cells, however, must be large. How? A neuron running down a giraffe's neck. Can you imagine a giraffe's neck? And there is a neuron from its brain running down to its leg. It may be several meters long. So for instance, but it is not shaped like a cube or a sphere. Instead, it is extremely long and thin. Why? This structure gives the neuron a large surface area with a relatively increase in volume. To maintain a suitable cell size, growth and division must be coordinated. If a cell more than doubled its size before dividing, before dividing, the cell in G2 grows larger. It doubles its size. But if the cell more than doubled its size before dividing, so the daughter cells would be larger than the original cell. In this case, if this happened, with each generation, cells would quickly become too large to live. But what about the other case? If the cells didn't double its size before dividing, so the daughter cells would be smaller than the original one. If this happened with each generation, cells would become too small to live. So this is why the cells have an upper and lower cell size limit. Thank you for watching this episode. Hope you have enjoyed and understood. See you in the next one. Thank you guys and goodbye.